yesterday, Ilsa sent me a message that was like, "Oh, cool talk. Could you present my talk? Because I thought Lauren was going to be able to do it, but now with the time shuffle, she's somewhere else." And I was like, "Oh, okay, I'll look and see." And I didn't realize they were right after one another. But Ilsa is writing out the hurricane, having been directly impacted, so I figured this is the least I can do for Ilsa is try to deliver her talk. But what I'm going to ask from you all is to also help support. I might need some audience participation to help interpret some of the different graphs and plots. Because it's not necessarily my area of expertise. And then, hilariously, as I was looking through these slides, I didn't even realize Ilsa was actually giving this talk for someone else. So not only am I not Ilsa is not Leila, who is the postdoc who heroically did all of this work, and I guess originally was going to be presenting it. So my deepest apologies to Leila, and I'm going to do my best. So, starting out with the ecology of Acropora palmata, I think we all know that Acropora is really unique for the Caribbean because it serves as that foundational reef builder on the crust habitat. So those really high wave energy environments is where you find those classic big thickets of palmata. We know that it's threatened under the Endangered Species Act, and which is why we're all working very hard to try to restore it because it plays such a critical role in the function of our reef ecosystems. And from a prior study of Ilsa's, which I actually think was really great, what they did was try to figure out, okay, what's driving differences in performance of palmata, so actually very similar to what Holland's study was. Particularly, they were interested in supporting population diversity because down at the dry Tortugas, there is palmata left, but it's actually a remnant population. It's just a single clone of Acropora palmata that lives in the Tortugas, is doing really well, but it's basically a genetic dead end. And there's a big concern about okay, how do we rescue this remnant population? Would it be possible to sort of transplant individuals in, essentially doing an assisted migration to see can we rescue what once was a very important population? So what they did was translocate, again, 5 genotypes. They had 5 genotypes of Acropora palmata. They were sourced from the CRF nursery, which is in the Upper Keys somewhere. I think these were the original sources of the genotypes. Yes, so they all originally came from an Upper Keys location, but they were maintained in the CRF nursery. And then they were outplanted to each of these 5 sites, going from a couple sites in the Tortugas all the way up the Keys, Lower, Middle, and Upper Keys. Transplanted out in replicates, and Ilsa was looking at various different growth metrics. And similarly, she published this already. You can read this in her 2020 publication, but they also found really strong site effects on survival. So what they found was site really mattered for both growth and survival, and where they saw the biggest growth and survival was actually down in the Tortugas. So if you look, these are the Tortuga sites, and I think this is Lower, Middle, Upper Keys. Makes a pretty nice correlation where you're seeing this highest calcification rate in every coral that went down to that Tortuga site. So all the transplants actually really loved being at that Tortuga site. And this study was trying to figure out why. Why is the Tortugas this amazing place for palmata to grow, even though we only have this one genotype left? So Ilsa had done this original study, and then this collaboration was started with the Gertoli lab, and Leila came on as a postdoc to answer these questions. And they did a whole bunch of sampling to try to solve this question. They were looking at various aspects of seawater biogeochemistry, so particulate organic carbon, dissolved organic carbon. They were also looking at microbial communities and zooplankton, because one of the ideas was okay, maybe there's some additional food sources in there, so we should be looking at what is in the plankton community, as well as doing some fun stable isotope stuff to try to see if they could trace whether it was heterotrophy or autotrophy maybe driving that. In addition, they did all of this other physiology to say okay, we see them growing better, but in addition to calcification, how are these corals feeling across these different reef sites? So biomass, chlorophyll A, symbiont densities, which thankfully some of the talks before the break introduced these as being fairly core metrics of host physiology, this heterotrophy component, as well as looking at protein and lipid content. So thinking about like energetic status of these coral fragments. What they found was perhaps not surprisingly, that these dry Tortugas corals, so again, remember this is the sites where they were seeing the highest calcification and the highest survival. They had significantly higher chlorophyll content, significantly higher symbiont densities. So these sites were seeing that. And that wasn't a negative effect, at least in terms of the context of this study, because they also saw that these corals had higher energetic reserves. So in addition to the higher symbiont, they were also seeing increases in protein content and total lipids, but also there was a bigger gradient here in the actual Florida Keys in terms of still seeing like higher lipid content as you're coming down into the lower Keys sites relative to the upper Keys sites. Okay, this is where I'm going to need somebody with lipid expertise to jump in. Dusty.
So my student is Heister, is doing a dissertation looking at lipid classes, and so Elsa and Leila and Andrea approached my group to see if we could identify some of these lipid classes. So we looked at storage and structural lipids. So wax esters, triglycerides are common storage lipids, and then sterols and phospholipids and AMPLs are common structural lipids. And so we used that to compare the populations, and we found some differences.
Thank you. How's that? There you go.
So yeah, long story short, the dry Tortugas populations had more of these larger lipid compounds, and I think we're interpreting that as maybe having a source of heterotrophy. There's an upwelling in the dry Tortugas, which kind of trying to put that together. And sterols, I've never seen this talk, by the way.
You're doing great. This is like, we've never practiced this either, but we're on it.
So sterols are an indication. It's like a cholesterol. And so if you're eating more zooplankton, you're going to have more cholesterol, so you'll have higher sterols. And so the story seems to fit for feeding on more heterotrophic sources.
Okay, yeah. And then this one.
Okay.
Yeah.
I'm not sure about the phosphorus.
Okay, cool. But similar, right? It seems to be they're fatter and happier down in the dry Tortugas. So putting all of this together, like there is a really big gradation in terms of like the coral physiology as you go from west to east, or sort of like the dry Tortugas up the gradient of the Keys, in terms of energetic status and other aspects of coral physiology that might explain those growth differences, might explain those survivorship differences in terms of the outplant performance. Again, similar to the calcification data. For the microbial communities, there also appeared to be differences, but it seems like this is interesting here, so I'm totally interpreting this without knowing what they necessarily want to say. But when I looked at this before, I was like, oh, that's really neat. That actually there seems to be kind of a big split between the two dry Tortugas sites, even though like for all the other physiological data, those are really similar. So I'm not sure what's going on there. This is the seawater controls mostly. And then this is a lot of like the rest of the upper Keys. So there might be some interesting differences in microbial communities that they're going to follow up on, but there's not another slide for that. So basically we're seeing in this study, genotype is not the biggest driver of performance of these palmata outplants. It really is these like site-specific characteristics, and a lot of it seems to point towards better condition. They do have data on the zooplankton abundance in terms of trying to link this better condition to the availability of food, or sort of like some of the ecosystem processes. They note that this was actually only a single time point, so this was just sampling at a single night. So even though it looks like there might be differences in abundance, like the community differences maybe aren't all that big, but they're not sure. Like they would need more nights of sampling to be able to see that. And it looks like there might be indications of upwelling. So in terms of this like dip in temperature, that might be facilitating greater production in that region in terms of like generating zooplankton content. Anyway, so I advocating here for thinking about the Tortugas as more of an opportunity to kind of reestablish a population where the coral are going to do really well, right, in terms of like growth and survival, and maybe become this really nice spawning population, because that might help seed the rest of the Keys because of the way that the prevailing current system goes. So if you establish a population here in the Tortugas, that could serve as like a nice source of larvae for the rest of the Keys. So sort of put them in the place that they're going to do really well. Okay, these are all the people that Leila would like to thank. Yes.
We do have a couple of minutes, but I don't think it's fair to fire any questions to Carly. But Dusty's here. I think Dusty's happy to entertain any questions from the room. If anyone has any previous questions, perhaps from Carly's presentation on behalf of Holland.